Thank you very much for this kind introduction. Despite my poor French, I understood some of it. And I hope to have an opportunity to thank this great Academy and the colleagues later in the day. And I understandably am extremely appreciative of this great honor. My laboratory was recognized for discovering back in the 1980s, more than 2 decades ago, the biology of the ubiquitin system. At that time, it was an obscure little thing. Very few labs, maybe 2 or 3 at most, were studying it anywhere in the world. Now it's actually one of the largest fields in modern biomedicine. Indirectly or directly, ubiquitin is encountered by almost 1/3 of the laboratories studying modern biology. And the reason for that is that ubiquitin system basically does everything that a living cell does. There are very few processes of any significance whatsoever that actually avoid using ubiquitin system. In fact, I'm not even aware of one. And it is for that reason, of course, that we're not going to do justice to the subject today if we shall discuss the ubiquitin system, because that would mean discussing the entire biology of today. So what I will try to do instead is to introduce to you the basic nuts and bolts of the ubiquitin system, something that we worked many years ago on, and then focus on more recent work that we have done about a subset of the ubiquitin system that I found over the years the most fascinating one to myself and to people who work with me. We call it the N-end rule pathway, and you will see shortly what it means. In eukaryotes, the N-end rule pathway is a part of the ubiquitin system, but remarkably, and we won't have time to return to this subject again, so I will mention it only now, the N-end rule pathway is also present in bacteria, in prokaryotes, where the ubiquitin system, the bona fide ubiquitin system, doesn't exist. Nonetheless, the N-end rule pathway, as you will see, is present in bacteria as well. So in some still ill-defined sense, because evolution of the ubiquitin system is shrouded in mystery even today, the N-end rule pathway is more ancient than the ubiquitin system. And with that small preface, let us begin by simply mentioning that degradation in general, particularly regulated degradation, plays a major role in life at many different levels. This is what we call regulated degradation, and by that simply is meant that the half-life of whatever biopolymer, big DNA, large precursors of messenger RNA itself, proteins, the half-life of these biopolymers varies as a function of time. It's not always fixed in the cell because it's highly regulated. And what we shall focus today on is a part of degradation, a very important part, that is played in pathways that target and selectively destroy specific proteins. But we should not forget that messenger RNA is also targeted and selectively destroyed. And even the precursors of messenger RNA are selectively targeted and destroyed. And even DNA itself turns over, especially when it is damaged. So a lot of this discussion about regulated degradation, by focusing on protein, would deliberately ignore a lot of interesting things happening to degradation elsewhere. And now we come to the ubiquitin system. It looks deceptively simple, even though it's one of the most complex systems in the cell. And I will tell you in a second why it looks deceptively simple while being so complex. So what is it actually? In eukaryotes, because bacteria don't have a bona fide ubiquitin system, they have bits and parts of it, but not the whole thing. A small protein, it's only 76 residues long, highly conserved among all eukaryotes, yeast ubiquitin and human ubiquitin are virtually identical, is consisting of this part and parts where ubiquitin is joined, covalently joined, to other proteins. What these other proteins are enzymes of ubiquitin metabolism. And the reason for doing this complicated dances becomes clear only after a brief introduction. So what happens? Basically, ubiquitin exists in 2 fundamentally different states. One is free state, a free protein, a free small protein, fairly abundant in the cell. and the other a state of polyubiquitin or monoubiquitin, where this contraption called polyubiquitin chain is covalently joined to that yellow thing called substrate. Now, what is substrate? Substrate, and this is the remarkable thing about ubiquitin system, can be just about any protein in a cell. In fact, is just about any protein in a cell. How does it happen? How do you join ubiquitin covalently to other proteins? These enzymes of ubiquitin metabolism, and again this scheme looks deceptively simple, and I will tell you where complexity comes in. This enzyme, ubiquitin activating enzymes, activates ubiquitin using NTP, placing it in a thioester linkage, a high energy bond. So ubiquitin is now ready to be donated downstream, and these ubiquitin conjugating enzymes, with the help of this most complex part, denoted here by this green box called E3, ubiquitin ligase, then generate this polyubiquitin chain. Now, the complexity of the system comes from E3. A human genome encodes about 1,000 different distinct E3 ubiquitin ligases. Now, 1,000 different proteins, it's about 3% of the total capacity of the genome. It's a very, very large family of genes, one of the largest in the genome. There are more ubiquitin ligases, in fact, in us than there are phosphokinases, to give you an example. And it is precisely because these E3s are such a numerous family of proteins, each of them recognizing specific degradation signals on substrates, on downstream substrates, that ubiquitin system has this immense reach, because different E3 proteins, different ubiquitin ligases, recognize different features of proteins and attach this polyubiquitin chain to them. Now, why to do that in the first place? It turns out that there is also in every eukaryotic cell a complicated contraption called 26S proteasome. It's actually a very large particle, about the size of the large ribosomal subunit, and it consists of this yellow part. Most of it has been by now resolved to almost atomic resolution. It's fairly abundant. It's about 10% as abundant as ribosomes, so it's quite abundant in the cell. And this part is essentially a channel with scissors inside, so that polypeptide chain of a substrate protein is unfolded by the ATPases of these regulatory caps on this core proteasome particle. The protein is unfolded, and its polypeptide chain is fed into the channel of the proteasome, where it is cut into small peptides, and then they exit from the other side of the proteasome. It is this machine that actually makes the entire scheme so important. And what I will not mention in the rest of this discussion is that ubiquitin evolved other uses other than degradation. Back in the 1980s, we discovered the first non-proteolytic functions of ubiquitin, because it turns out that joining ubiquitin to a protein may not necessarily, not in all cases, confer on that protein a short half-life. But we will skip that entirely today. Now, one last thing that I should mention is one might ask, given all this discussion, even if you don't know anything about the ubiquitin system, I'm sure many people in the audience know quite a bit about it, but suppose you don't know anything about it and you followed what I said closely, the natural question to ask, given what I already told you, is how does the ubiquitin system know whether to put ubiquitin on a protein or not? Well, it turns out that all proteins carry or don't carry specific features. It could be anything. It could be conformational determinant. It could be a specific sequence in the protein. It could be anything you can imagine. And these things are called degradation signals, or as we suggested to call them many years ago, degrons. And these degrons is what attracts ubiquitin ligases to proteins, resulting in this polyubiquitin chain. So this is actually where all this complexity comes from, and this is why ubiquitin system has such pervasive influence on the rest of the cell. Now, by the end of the 1980s, when this picture was already there, and partly because of our work and partly because of the work of Avram Hershke, laboratory in Israel. I tried to see what else we can do to advance this field in a way that would be not only helpful in general, but also interesting, for example, to myself. And we focused on a pathway that we discovered exactly 30 years ago in the mid-1980s. Andreas Beckmeyer and Daniel Finley in our laboratory discovered a pathway that we call the N-degron pathway. And we shall come to it in just a second. And the technology, the technique that led to this pathway, called ubiquitin fusion technique, is shown on this slide. The pointer is not very strong. I hope you can still see it. In any case, I will just hand wave it. I think it's dying on me, actually, as we speak. And so the ubiquitin fusion technique, which is still even after 30 years, is still the method of choice by anybody who wants to do anything resembling things that I'm about to describe, is actually a very simple thing, as you see. It allows you to expose at the end of the protein any amino acid residue of your own choice. Now, why this is not a trivial thing to do otherwise? Well, then you have to recall a little bit of textbook molecular biology. All messenger RNAs carry AUG as the initiation codon, and it encodes the amino acid methionine. So all proteins start with methionine at the end. And then whatever happens to this methionine, it could be N-terminally acetylated, it could be taken off by specific enzymes. Thank you. Thank you very much. It could be taken off by specific enzymes called methionine aminopeptidases. Lots of things can happen to the N-terminal methionine in a protein, but you don't control it very well. So ubiquitin fusion technique basically converts the tricky and difficult to control process of methionine removal to a very different proposition, where you attach ubiquitin linearly by encoding it as a DNA fusion to a protein. And then it turns out there are many so-called deubiquitinating enzymes in every eukaryotic cells. Human cells, for example, contain up to 50 different ubiquitin specific deubiquitinating enzymes that have a common property of being able to cut here co-translationally and result in any desired residue that you wish. That technique was invented by Andreas Beckmeyer in the lab in 1986, 30 years ago. It's still the technique to use if you want to do something like that. And out came completely unexpected result, which we called, for the want of better word, the NN rule. And this is what you're looking at. The NN rule is basically a relation between the in vivo half-life of the protein and identity of its N-terminal residue. What emerged was basically several discoveries in one, thanks to the ubiquitin fusion technique and probably luck. And that is that, first of all, we learned what the nature of degradation signals in proteins is. We learned the identity of the first class of degradation signals, so degrons, or as we started to call them N-degrons, because they were N-terminal degradation signals. We also learned that, remarkably, in fact, we were amazed to see that a single amino acid residue at the end of the protein is sufficient to confer a short half-life on the protein. And the half-life varied over 4 orders of magnitude, over almost 10,000-fold. For example, if the protein has alanine residue at the end, the half-life of a test protein, it could be just about any protein you want, is about 30 hours or more, very long. And any of those residues at the N-terminus of the protein will confer a long half-life. However, if a protein bears arginine at the end, its half-life is almost unmeasurably short. Actually, it's even less than 2 minutes. And so it goes. You go from very long half-life to extremely short, depending on the identity of the residue at the N-terminus of the protein. Now, it took then at least 10 years, in fact, this work continues as I speak, to decipher the actual mechanics of things that make this table possible. And the analogy, for example, is the genetic code. The genetic code table consists of 64 cells, and it's actually a fairly simple table. But to implement that table in the cell takes almost half of cell's machinery. Very much the same obtains in the case of N-degron pathway, because what I'm showing you on the next slide is the current understanding obtained largely in our laboratory over the last 20 years of the mammalian N-degron pathway. We work both on yeast and mice, and this particular slide is about mammalian N-degron pathway. Yeast has exactly the same pathway, but slightly less elaborate. And I will just walk you quickly through this pathway because this pathway has tremendous number of functions, not as many as the ubiquitin system at large, because the pathway is a part of the ubiquitin system, but amazingly many. And we shall touch upon some of them as time goes by. But let me first to walk you through this pathway, because it looks complicated, but it's actually logically very simple. This little yellow oval is the rest of the protein molecule. And these single letter abbreviations are N-terminal residues of the protein. It turns out that the N-end rule is a hierarchic code. It is a code because it relates one thing to another. It, as mathematicians say, maps one thing to another. And there are three kinds of destabilizing residues in the N-end rule. We call them primary, secondary, and tertiary. In this particular case, primary destabilizing residues are those that are directly recognized, which simply means bound to, by ubiquitin ligases of the N-end rule pathway. You may recall the first slide that describes the nature of these E3 ubiquitin ligases. In you and me, there are several of them. In yeast, only one, making genetics of the N-end rule pathway much easier to study in yeast. But the principle is exactly the same. These ubiquitin ligases directly bind to these primary destabilizing residues, and what they are, leucine, phenylalanine, tryptophan, tyrosine, isoleucine, arginine, lysine, and histidine. Now, these are basic residues, 3 basic residues, and these are bulky hydrophobic residues. And these ubiquitin ligases are machines that, among many other qualities they have, these are large proteins, they also have specific binding sites. One binding site is specialized in binding N-terminal bulky hydrophobic residues, and the other binding site is specialized in binding basic residues. What happens then, once these residues are bound by ubiquitin ligase, that polyubiquitin chain that you saw on the earlier slide is built up on this yellow oval of the protein, and the protein is destroyed by short peptides. What is a secondary destabilizing residue? Secondary destabilizing residue is, by definition, something that ubiquitin ligases will not see, will not bind to. However, all eukaryotes, from yeast to us, have this very interesting enzyme called arginyl tRNA protein transferase, R transferase for short, encoded by AT1 gene. All of these genes, of course, have been cloned in the lab over many years. So what you are seeing on this slide is enormous condensation of many years of work by many people in the laboratory who actually proved that this heme works the way it looks on the slide. And this arginyl transferase enzyme uses arginyl tRNA that normally is used in translation. But instead of placing arginine as an emerging polypeptide chain on the ribosome does, at the C-terminus of the growing polypeptide chain, this enzyme takes arginyl and places it in front of the protein. So it arginulates N-terminal residue, and these are destabilizing N-terminal residues, oxidized cysteine, aspartic acid, and glutamic acid. These 3 residues will be recognized by arginyl transferase, and the protein will become 1 amino acid longer before it is destroyed. Why to do that? Because arginine is one of the primary destabilizing residues. Ubiquitin ligases would immediately be able to bind to this protein, and this dance that ends up with protein destruction will continue because of this ability of AT1 transferase to conjugate arginine to the end of the protein. What are tertiary destabilizing residues? Tertiary destabilizing residues, yet again by definition, are those that cannot be recognized either by arginyl transferase or by ubiquitin ligases, but they are recognized by yet another component of the N-end rule pathway. And these are new enzymes, entirely new enzymes that have been cloned in the lab, both in yeast and mammals over the years. And these enzymes are N-terminal amidases. They deamidate asparagine and they deamidate glutamine, and they do it if and only if these residues are N-terminal. They will not do it to N-internal asparagine or glutamine. The result, aspartic acid and glutamic acid. Aspartic acid and glutamic acid will be arginulated, and the protein will be eventually, through the intermediacy of these ubiquitin ligases, destroyed by the proteasome to short peptides. Cysteine is an interesting and very important biologically special case. Cysteine will not be recognized by arginal transferase. It will be only cysteine sulfonate or sulfinate that will be recognized. That is where the sulfur atom is oxidized by joining of two oxygen atoms, which looks a little bit like aspartic acid, and the enzyme is fooled to arginulate it. How that cysteine becomes oxidized cysteine? It turns out that both nitric oxide and oxygen are involved. And so the N-rule pathway emerged about 10 years ago as a new kind of sensor of both nitric oxide in the cell and oxygen, because this reaction requires both oxygen and nitric oxide. And we probably will not have time to touch it any further, and that's why I mentioned it. So you have this mechanistically complicated but logically very clean and clear system that enables any protein that carries these N-terminal residues to be destroyed in a regulated manner, because all of these enzymes are also under very heavy and rigorous control in the cell, both through expression of their genes and through other modifications of the enzymes themselves that vary their activity. Now, this is an example of the yeast N-rule pathway. What you just saw before was mammalian N-rule pathway, and I won't repeat description of the mammalian N-rule pathway over here, because basically what you see is a copy of it in yeast, but somewhat less elaborate. For example, notice that cysteine disappeared because yeast normally do not produce nitric oxide, and therefore cysteine doesn't get oxidized in yeast. Then in 2010, 6 years ago, a talented postdoc in the lab from Korea, Cheol-Sang Hwang, who now has his own laboratory in Korea and continues to study the N-rule pathway. In fact, the field of the N-rule pathway grew over the years, as often happens, through efforts, independent efforts by former postdocs in the lab. So there are now several, probably 5, if I recall, full-time N-rule laboratories, all of them started by former postdocs in the lab. And what he discovered back in the lab in 2010 is an entirely new N-rule pathway that nobody, including us, suspected exists. And I will very briefly mention this pathway because it's actually both very interesting and biologically important. We call it the AC N-rule pathway. You will see in a second why it is called now. And the old N-rule pathway, the one I mentioned to you just 2 minutes ago, is now called the ARG N-rule pathway by the presence of N-terminal arginulation in it, to distinguish it from this topologically very similar, because it involves N-terminal residue of the protein, but mechanistically entirely distinct AC N-rule pathway. What is AC N-rule pathway? Notice that these N-terminal, again, this little oval is the rest of the protein, right? And these are N-terminal residues. And if you look, these letters are large enough probably to see clearly. And if you look carefully, what you will see that these amino acids, with exception of cysteine, actually in yeast even without that exception, because cysteine is not present here, they don't actually coincide with any of those residues. There is no overlap, so to speak, between those two things. Why so? Well, it turns out that none of these residues, none, literally none, will be normally exposed when the polypeptide chain emerges from the ribosome. Methionine aminopeptidases will cleave methionine of the emerging polypeptide chain off if and only if the second residue to become N-terminal after removal of methionine is not any one of these things. Not leucine, not lysine, not aspartic acid, not glutamine. All of these residues will forbid removal of the methionine. So how is it that these residues become exposed at the N-terminus in the first place? Other proteases do it. Other endoproteases, after the protein is fully made, becomes mature, and the list of these proteases is long and still growing. You know, most of them probably know the names of these proteases. These are caspases that control programmed cell death, apoptosis. they can create these N-terminal residues, and they do, and therefore the N and rule pathway is a major, it emerged, as we showed several years ago, as a major regulator of programmed cell death, or apoptosis, as it is also called. And other proteases like calpains, for example, neuroscientists know about calpains very well because these are calcium-activated proteases that become activated after calcium transients as a result of action potential impulse arriving at the gap between axon and the postsynaptic neuron result in calcium fluxes into the neuron and activate calpains, and they begin to create N and rule substrates of this pathway. This one is completely different. Why? Because these residues are small. Alanine, valine, serine, threonine, and cysteine, and glycine and proline are small enough to allow methionine aminopeptidases to remove methionine without any problem, co-translationally. Now, for a very long time, both us and everybody else believed, turned out to be completely erroneously, this modification, N-terminal acetylation of the alpha amino group in the protein, was the oldest modification of the protein ever discovered 65 years ago. And ever since, it was biologically enigmatic modification. Why? Because, and again, I'm not going to do justice to the subject because we won't have time for it, but acetylation recently emerged as one of the major protein modifications, akin to phosphorylation of proteins. However, the bulk of acetylation happens on internal lysine of proteins, not on N-terminal residue. So N-terminal acetylation of proteins is a very different beast, distinct from internal acetylation, which is highly reversible. The acetylases and deacetylases, the acetyl group is continuously taken off and put on lysine residues and involves a lot of interesting biology that we're not going to discuss today because this is a very different affair. This acetyl group, to the best of anybody's knowledge, is placed irreversibly on the protein, co-translationally by N-terminal acetylases, specific class of enzymes hugging the ribosome and doing it co-translationally, and then stays on the protein for the rest of its lifetime in the cell. That was all very well known before us, and everybody and their mother, including us, believed that one function of the acetyl group at the N-terminus of the protein is to protect the proteins from the N and rule pathway. Well, just the opposite turned out to be true. It turns out, and for now we understand why this discovery was kind of post-mature, why it was overlooked for many years. We understand it, but I won't have time to describe why exactly it was overlooked for so long. But what actually happens is that when these N-terminal residues are co-translationally acetylated, there are specific ubiquitin ligases in the cell, different from ubiquitin ligases of the arginine and rule pathway, that will actually recognize N-terminal acetyl as a degron, as what we call N-degron, and destroy them to short peptides in the same regulated manner. So there are now two different N and rule pathways, and if you see among them, they encompass the entire genetic code, just about the entire genetic code, all 20 amino acids in different proteins under certain conditions and in certain contexts can become destabilizing. This is actually just a glimpse of understanding about how non-trivial, technically interesting, but mechanistically complex are the process of recognizing N-degrons. N-degrons, as everything in biology, things begin to look simple and then stop looking simple once you begin to understand them really much better. And the same is true of N-degrons. N-degrons only seem to be simple, a single residue in the protein. But in order for the protein to be actually degraded, a polyubiquitin chain has to be placed somewhere in the protein, in this case on internal lysine. And so quality, quote unquote, quality of that lysine, its ability to become spatially close to the N-terminal residue becomes very important. And the final configuration, crudely depicted here, is when polyubiquitin chain is captured by the proteasome. Proteasome is kind of deliberately dumb. It doesn't pay attention to what the protein is. It doesn't pay attention to the nature of N-terminal residue. It only pays attention to a simple presence of polyubiquitin chain in the protein, allowing, therefore, a distribution of the intelligence of the ubiquitin system into many different pathways that all converge on that funnel downstream called the proteasome. And this is what happens. The loop of the protein enters the channel in the proteasome, and the scissors begin to work inside the channel. And I will not discuss these N-degrons in greater detail, but what you see, of course, they have all kinds of interesting mechanistic properties because we will never finish otherwise. And what I wanted to mention and what is shown on this slide is a statement that explains why we are not going to do justice today to even the N-rule pathway, let alone the ubiquitin system. Some of those functions of the N-rule pathway are understood mechanistically, not only functionally as well. But there are so many of them by now. In fact, I don't even remember all of them without looking at the list, which is really amazing because most of them were discovered in our lab. That even briefly mentioning all of them in the space of an hour would be a problem, while a comprehensive discussion of them is plainly impossible. And therefore, an unavoidable compromise is to give you a little glimpse of some of these functions, but to dispense with the rest of them. And the next slide, I think you don't have to read it because it's largely for me, because otherwise I won't remember all these functions, is just to list some of the functions. In fact, this list is incomplete. These are functions of just Argen and rule pathway primarily, and we shall discuss ACN and rule pathway, the more recently discovered pathway, in just a little greater detail in the remaining few minutes that I have. So the Argen and rule pathway, as I already told you, has emerged as a new sense of nitric oxide and oxygen. It has emerged as a new sense of heme, or rather hemin, which is ferric oxide, ferrin 3+, heme, because hemin amazingly physically binds both to arginine transferase and to ubiquitin ligases of the N-rule pathway, and regulates them in very interesting ways that are beginning to be understood. And so it's actually a new sense of heme in the cell. As I said, senses nitric oxide and oxygen. It senses short peptides. Transport of short peptides in cells turns out to be regulated in very interesting ways, which we won't have time to discuss today, by the N-rule pathway. And some of you know that short peptides, of course, are extremely important, at least in one arena. Well, they're important as food for microorganisms, but they're also important for us, because it is short peptides that are bound to MHC proteins, major histocompatibility complex proteins, and it is these short peptides that are presented to T cells. And it is because of that we're able to sit here and enjoy their relatively pathogen-free existence, because otherwise they will eat us alive. And therefore, it is these short peptides that allow immune system to distinguish healthy cell from infected cell, for example. And part of the transport of the peptides is regulated by the N-rule pathway. As I already mentioned, the N-rule pathway has emerged in the last 6-7 years as a major regulator of programmed cell death, apoptosis, by destroying proteins that get cleaved and become active in regulating apoptosis positively. So by destroying these proteins, the N-rule pathway buffers cells against apoptosis so that only very strong proapoptotic signal will be able to overcome this resistance by the N-rule pathway and push cell beyond the point of no return where it will be actually killed. And so this is another function of the N-rule pathway. Amazingly, yet another function emerged as a result of recent work by Chris Brower and other people in our lab, and that's the N-rule pathway, specifically the N-rule pathway, appears to repress neurodegeneration. And it does it because, as many of you know, neurodegeneration is caused, at least in part, in a very large part, by all kinds of problems that attend to aggregation propensity of a variety of different proteins, some of them normal, some of them mutant, some of them emerging as damaged during aging, for example, even during normal aging. And all these problems very often involve generation of fragments of proteins. And these fragments sometimes are much more dangerous than full-length proteins because they are more prone to aggregation. And it turns out that the N-rule pathway very often catches these fragments and destroys them. So the N-rule pathway is now emerging as a major bulwark against neurodegeneration. And so there are all kinds of other things, and not to lose any more time, I just, this is the story about apoptosis, but since I'm slowly, not so slowly actually, running out of time, this is the scheme through which N-rule pathway controls apoptosis. It basically, as I already told you without showing the actual picture, destroys proapoptotic protein fragments and therefore buffers cell against onslaught of proapoptotic signaling that occurs when cells may enter apoptosis unless protected by something. That something, among other things, is the N-rule pathway. And then we come to the mammalian N-rule pathway, and this is just a very brief, since I don't have much time left, this is a very brief illustration of the importance of the N-rule pathway in developmental biology. Because developmental biology, as many of you know, involves ubiquitin system in a great variety, in major ways. And to give you an example of what happens when you begin to tinker with N-rule pathway, for example, if you delete in mice, as we did some years ago, and we continue to work on this, if you delete arginine transferase gene, which luckily happens to be a single gene, and therefore the phenotype is completely penetrant, well, if you delete it in an egg, the animal is not born. And if you delete it in the adult mouse, there are all kinds of interesting effects. If you delete ubiquitin ligases of the N-rule pathway, if you delete, for example, just those two, again, it's embryonic lethal because it's incompatible with life. And this is something that, for example, to give you an idea of how important the N-rule pathway is in development, this is what happens when you delete only two. So these are mouse double mutants, and it usually takes trouble to produce double mutants even today with CRISPR and all the other technologies that makes it easier. It will become a little faster. But if you have these double mutants in mice that don't have UBR1 and UBR2, two major ubiquitin ligases of the N-rule pathway, they still have the N-rule pathway because the other ubiquitin ligases are there. But it's weakened and kind of different because each of them is similar to the other one, but not really identical. What you see, for example, is enormous catastrophe, neurological catastrophe that attends the development, for example, of the forebrain. The forebrain is already beginning to be underdeveloped by embryonic day 10, and by embryonic day 11, only a day later, you have normal forebrain in this embryo developing, and complete catastrophe and resorption of the forebrain in the 11-day embryo lacking those two genes. So this is just one of many different illustrations of the importance of this pathway in development. And the last thing I was about to tell you is the importance of this new N-rule pathway, which we call the AC N-rule pathway. It has many functions, and in fact, we are fairly confident that we don't know many of those functions yet. They will continue to emerge. But one of them that already emerged is quality control. Basically, what happens, and this is the last thing I'm going to say, what happens is you have a protein here, P1, we just call it, let's say, P1, and it has acetyl group placed at the N-terminus. It now has two fates, two competing fates available to it. One is to become bound to the ubiquitin ligase, we call it N-recognins, of the AC and N-rule pathway, and to be destroyed to short peptides. The other fate, however, is to bind to a protecting protein. What is protecting protein? Let's call it P2. It's any protein that binds it as a part of cognate protein-protein complex, and it binds it, interestingly, in such a way that this AC group no longer is available for attack by N-recognin. So this dichotomy allows you to control protein quality. Why? Because if the protein emerges and doesn't find an abode, a cave, to hide its N-terminal acetyl group, it will be simply destroyed. So this system, without knowing what it is doing actually, can control quality of the protein by simply demanding that once you are born, you have to find a place to hide your ant as quickly as possible, or else you will be destroyed. And that's how it works. And the picture, I will skip this slide since we're running out of time, but the picture that illustrates this remarkable fact is it's a crystallographic, high-resolution crystallographic. It's actually atomic resolution picture, but here the ribbon diagram is used. Was created in the lab of David Barford, who studies another ubiquitin ligase. And what you see, I hope you can see this. So this is one protein, Cat9, and I will skip all details because it really doesn't matter what proteins are here. What matters is how do you hide your ant, okay? And this is another protein shown here is a skeletal structure. And what you see, N-terminally acetylated methionine of this protein is hiding deep in the chamber of this protein. And so N-recognin of the AC and N-rule pathway cannot reach it here. And that's how these complexes form for many, many different proteins. And so proteins, in other words, evolved not only to bind each other, but they also evolved to bind each other in such a way that they protect each other from attacks of otherwise continuously active proteolytic systems that would destroy this protein unless its N-end becomes sterically inaccessible. And these are examples of biochemical proof of what I just said. We shall skip that. And what we have seen are very few, relatively few, as many as can be discussed in such a short time, examples of this enormous variety of functions that even a corner, not just the entire ubiquitin system, but a corner of the ubiquitin system called the N-rule pathway has. You can easily imagine in your mind's eye, if this is that many functions that a corner of the ubiquitin system has, you can imagine how many functions of different kinds the entire ubiquitin system has. And there are many people, some former members of the lab are listed here, and among them are contemporary leaders in the ubiquitin field. Several of these people are now having, quite a number actually, having their own laboratories all over the world and leading research on the ubiquitin system. And these are people in the lab that work together with me as we speak. And I thank you very much for your attention.
Thank you for this very nice presentation. I'm sure there will be questions. Who want to start? Moshe Yeti.
Alex, I'm on your back.
Yeah.
If I remember correctly, ribosomal proteins and histones, if they don't find the partners, are
Yes.
degraded. Does it work?
Yeah, that's an excellent question. In fact, what Moshe Yaniv asked is how does this system or any other system of that sort controls protein stoichiometry in very large protein complexes? For example, ribosomes have dozens of different proteins, and no matter how precisely you control gene expression, I don't know how to stand. No matter how precisely you control gene expression, you will always end up with incompletely coordinated, balanced stoichiometry of proteins entering the system and building up either ribosome or proteasome or any other multi-subunit complex. What to do? Well, cells adopted a very simple strategy. The idea goes back a long time, but degrons involved have not been known. We have preliminary evidence that AC N-degrons, that is N-terminal acetylation, plays a role in controlling input stoichiometry, for example, of ribosomal proteins, because 3/4 of ribosomal proteins are N-terminally acetylated. And when you look at crystal structure of the ribosome, most of these proteins have the AC group buried. So while they're free, they're vulnerable. The moment they build themselves into the ribosome, they're invulnerable.
Another question. As I remember, SV40,
Yes.
VP1 is also acetylated.
Yes, well, and so it is conceivable. That has never been studied, I think. But what you imply by this question is that it probably is also under regulation of this I would not be surprised.
Pascal Cossard.
Okay, so you said that you were going to talk about the NN rule and then talk about how the ligase is looking for the site of ubiquitination.
Yes.
So you have this recognition site in the N-terminus, and
Right.
my question is, okay, you may have a very close lysine, which
Right.
will be ubiquitinated, but very often you have also sites very far away in the
Yes.
protein. So can you comment on that? How?
Yes. In fact, instead of showing slide, I will just hand wave it. The essence of it, the details, the devil is in details, of course, but the essence of it is actually quite simple. Imagine in your mind's eye that you have an N-degron, which means N-terminal residue of the protein, but the complete N-degron is more than that. It's actually at least 2 determinant signal, as you just mentioned, because it also contains N-terminal lysine. Ubiquitin ligase, it's all Brownian motion and diffusion in the crowded environment in vivo in the cell. The ubiquitin ligase collides at random, of course, with N-terminal residue. There is affinity. It sticks to this residue. And now the race for time begins. Because piggybacking, I didn't mention it explicitly, piggybacking on this ubiquitin ligase is this other contraption called ubiquitin conjugating enzyme, typically a relatively small enzyme that actually positions ubiquitin on a lysine. And now this ubiquitin conjugating enzyme is frantically looking for a lysine. And that enzyme has only so much time to find the lysine before the ubiquitin ligase proper will dissociate from N-terminal residue. So it's a kinetic problem that has to be solved in real time. What happens? As you mentioned, if lysine is very close to the N-terminus, the search for that lysine ends very quickly, and the protein is rapidly destroyed. If the lysine is very far down the road in the polypeptide chain, and if the protein is not folded closely to the N-terminus, that lysine will never be targeted, and eventually N-recognin, ubiquitin ligase, will dissociate from the N-end, and the protein will never even know that it was in danger. However, because proteins are folded in all kinds of intricate ways, they evolved, especially those proteins that evolved to be short-lived. Not just damaged proteins, but proteins that have to be short-lived because they're regulatory proteins. They have their lysines positioned in space in such a way that this piggybacking ubiquitin conjugating enzyme can actually find them in time. And it doesn't have to be very close along polypeptide chain as long as the folding of the protein brings it close to the N-terminus. Such examples are already known. And that's a partial answer to your question.
Yeah, because sometimes you have several sites.
Exactly, and very often ubiquitin system at large, not just the N-and-rule pathway, but other pathways of the ubiquitin system, there are easily several hundred of different pathways of the ubiquitin system. We're talking about something so immense that even mentioning it takes more time than we have. So all of these things sometimes have alternative lysines and can choose between them at different transition probabilities.
Yeah, and if you mutate one, then
Yes,
the other
exactly,
one will be.
exactly, there is a redundancy. Yes.
Thomas Lecuy, ensuite Eric Westhoff.
So I was curious, can you tell us a little bit about the core system that exists in prokaryotes and what similarities?
Well, it's another interesting question, and one can easily give another seminar about it. The bacterial N-and-rule pathway is a curious beast because the general scheme of the bacteria, I didn't even have slides of the bacterial N-and-rule. We discovered N-and-rule pathway in bacteria in 1991, 5 years after finding it in eukaryotes. And what we found was an interesting thing, because first of all, there is no ubiquitin in bacteria, but there are proteasome-like proteases. One of them is called CLIP-AP, and it happens to be the protease that destroys N-and-rule substrates. How does it find it? How does it find an N-and-rule substrate in E. coli? It turns out that the N-and-rule in E. coli is a simpler version of a eukaryotic N-and-rule. It has 4 bulky hydrophobic residues, like leucine, phenylalanine, tryptophan, and tyrosine, and it has 2 basic residues, arginine and lysine. That's it. All other residues will be stabilizing, quote unquote, in the E. coli N rule. Who finds these N-terminal residues? A small protein called ClipS. It's an adapter protein for ClipAP protease, proteasome-like ClipAP protease. So it binds just like these very large ubiquitin ligases of eukaryotes. It binds to N-terminal residues of these N-rule substrates in E. coli and physically brings them to the ClipAP protease, where the process of destruction of the kind I described then ensues, and it doesn't require ubiquitin. It's actually kind of posing a problem, if you will, because E. coli are just as sophisticated as us in some sense, because they, in fact, one could even argue that they are more evolved than us because they had time to try many more constructions per unit time given their short generation time. It's an interesting argument whether eukaryotes are more evolved or less evolved compared to seemingly simple creatures, and the argument that they are less evolved is actually fairly strong. So how does the N-rule pathway in E. coli manage without ubiquitin? Part of the answer is very high processivity of ClipAP protease, and the other part of the answer is that proteins in E. coli that actually are subject to degradation by the N-rule pathway are, crudely speaking, looser. That is, they don't require as much unfolding effort by ATPases as typical eukaryotic proteins do. And it's a partial answer to your question.
Yes, merci. Thank you for your presentation. So you said that the R transferase is an amino acid tRNA synthetase.
Yeah.
So how can you delete it without impairing translation, normal translation?
Well, actually, it's not. So let me clarify it a little bit. I'm glad you asked the question. There is arginine tRNA synthetase.
Yeah.
What it does, it takes amino acid arginine, for example, right, and joins it to tRNA, forming arginine tRNA. This enzyme is different. This is not arginine tRNA synthetase. This enzyme depends on the arginine tRNA synthetase to supply it with arginine tRNA.
Okay.
Then it takes arginine tRNA and the high energy moiety of arginine and covalently conjugates arginine to another protein. So it's a completely different enzyme, but it does work
On the same
in concert with the enzyme that you mentioned.
All right, these two steps.
Yeah.
Thank you. And then also, how is it with mitochondria?
This is actually a fascinating question because I was always, since mitochondria, of course, are remote descendants of bacteria, you would expect them to also have the N-rule pathway. And we tried for quite a long time to find it for all kinds of reasons that I will skip. We were unsuccessful. But then Nicholas Pfanner in Germany, 3 or 4 years ago, reported fascinating evidence suggesting, but not yet proving, that mitochondria, the inner matrix of mitochondria, also has its own separate N-rule pathway. The work continues as I speak, but they didn't publish it yet. It probably does have it.
Claude Desplein, ensuite je reviens vers vous.
So one common theme of your talk was actually the fact that everything is irreversible. And yet you have the deubiquitination enzyme.
Yes.
So what do they do exactly?
So deubiquitinases are a complex addition to this whole thing. And there are many different functions of deubiquitinases. And again, for reasons I mentioned, I won't be able to do justice in answering your question in full. So I will just give you a conical section of the answer. That partial answer basically consists of 2 parts. One is that the major function of deubiquitinases, if you don't talk about very specific functions, because some of them have very specific functions, is editing. Because ubiquitin system at large, and the N-rule pathway being an example of ubiquitin system, makes mistakes. These things are not perfect. Sometimes degrons are engaged in a way that doesn't work very well, the system jams. Sometimes the protein is delivered to the proteasome, but ATPases of the proteasome lock in an unfavorable configuration. In fact, such examples have been uncovered, where the protein basically makes this proteasome no longer functional. It's stuck. It cannot enter the channel, and it cannot easily dissociate because polyubiquitin chain keeps it in place, because proteasome has strong affinity for polyubiquitin chain. What to do? You wasted a very complicated particle like proteasome. It is in this case, and many others like it, that deubiquitinases free the protein to swim away from the proteasome by removing polyubiquitin chain. And this process, competition between building up the polyubiquitin chain and removing it, goes on all the time. So that's answer number one. And very quickly, answer number two is that because you and I have more than 50 different deubiquitinases, all of which have a common property of recognizing ubiquitin and cleaving immediately after residue 76, but otherwise they have other properties. And one of them is being able to recognize specific proteins. So it has a domain that is deubiquitinating, but has other domain that, for example, binds to protein like p53, a well-known tumor suppressor. And so these deubiquitinases may keep the protein stable despite it being attacked by the ubiquitin system by constantly removing polyubiquitin that is essentially creating a futile cycle, creating a substrate cycle. And there are known deubiquitinases that are specific for several different proteins, not just for any protein. And these are two major functions of deubiquitinases.
S'il vous plaît, monsieur, ensuite, madame.
Thank you very much. I'm Jozo Delic from CUA, and I am wondering whether N-enroll pathway is involved in simulation and nedylation that are...
I understand what the question, and I will just introduce a little bit what you just said, because I have to decode what you said. Yeah. Nedylation and simulation refers to reactions. What I didn't mention during the talk was that after ubiquitin system came into being, approximately, we also contributed a bit to this process 15, 20 years ago. After ubiquitin system came into being a long time ago, variety of labs, including us, discovered that, remarkably, they're so-called ubiquitin-like proteins. By now, the count stands at about 10 or 12 in mammals and about 7 or 6, for example, in yeast. These proteins have all kinds of accidental names, RAB1, SUMA, NED8, and reactions that these proteins undergo are called nedylation, sumylation, and these are fancy words that denote basically the same thing as ubiquitination. That is, the C-terminal residue of these proteins, SUMA, NED8, get covalently joined to lysine residues, typically lysine residues, there are some exceptions, of other proteins. What happens when this happens? What actually happens? In some relatively rare, but now already known cases, these ubiquitin-like proteins can also signal proteolysis. But the major function of these other ubiquitin-like systems is to control other things in a non-proteolytic manner. One good example are two ubiquitin-like proteins that control autophagy, for example. Not NED8 and not SUMA, but other ubiquitin-like proteins, they get conjugated to regulatory proteins that control autophagy, which is another major proteolytic systems in all eukaryotic cells, and this gives you an idea. Now, the N-enroll pathway, we're virtually certain, is regulated by both nedylation and sumylation, but over the last 30 years, we never found the time to actually investigate it. There are strong hints that they are, but it's not known in mechanistic detail.
Madame, et ensuite Daniel Louvard.
What can be the efficiency of ubiquitination? What can be the efficiency of ubiquitination of cytoplasmic located acetylated protein by the DOA 10 ER located E3?
Yes. Well, to answer that question, I have to mention something that I simply neglected to mention before. So, for example, it looks odd if you look at the scheme of the N-enroll pathway. How can you actually have an efficient proteolytic pathway if the mechanistic dance before you actually destroy the protein is so complicated? But amazingly, these machines are very efficient. If you design your N-degron properly, and I will return to your specific question in just a second, if you design your N-degron properly, for example, by placing this glutamine in a right way in conjunction with a specific lysine in the protein, you can fall from here to nothing, to short peptides, in less than 2 minutes. So this is actually a very efficient pathway, despite the fact that it involves several different steps that have nothing to do with proteolysis as such. And so the same applies to AC N-degron pathway. We already know that half-life of a protein that is N-terminally acetylated and, for example, targeted by DO10, ubiquitin ligase that you mentioned, can be as short as a minute or less. And so these are very efficient pathways.
I'm here on this side.
Yeah.
Right? Okay. I think you partly answered the question that I wanted to address for a long time, Mr. President. How much the subcellular localization of the substrate and the machinery contribute to this already complex and sophisticated regulation of protein stability?
Right. The short answer, of course, it's very important to be in proximity to things of that sort, because diffusion of these very large complexes limits kinetics of targeting in significant ways. A good example is actually provided by the preceding question, because DO10, ubiquitin ligase, remarkably, is not a quote-unquote soluble cytosolic protein. It's actually many spanning transmembrane protein that sits in the endoplasmic reticulum membrane with its active site facing the cytosol. So it's surprising that such an enzyme evolved the ability to recognize N-terminal acetyl group and target soluble cytosolic protein. The partial answer is that it doesn't do only that. We already know it. The partial answer is that DO10, as you know, if you are transported into the lumen of endoplasmic reticulum, and if you are not behaving there sufficiently well, for example, refuse to fold quickly, or cannot be glycosylated, as ER does to proteins, sufficiently well, you are declared misfolded or misglycosylated, and usually what that entails is exile back into the cytosol, retrotranslocation as they call it, and this retrotranslocation results in destruction of this previously ER protein by the ubiquitin system. What we are now discovering is that DO10 is involved in retrotranslocation of these proteins, but in addition, the recognition very often involves anti-acetyl group. So maybe evolutionary pressure that drove for this strange contraption of having an enzyme that is ubiquitin ligase sitting in the plane of membrane instead of being freely diffusing has to do with this dual function of DO10, targeting both soluble proteins that swim by and targeting proteins that are ejected from endoplasmic reticulum and have to be degraded in the same manner. It's a partial answer to your question.
I will take the last question. Ah non, allez-y, avant-dernière.
Thierry Menel, So my question is about a mechanism which has been described, which involves amino-terminal ligation of ubiquitin to some short-lived proteins, such as transcription factors like MyOD. So how do you integrate this kind of degradation in your general model?
That's another interesting question for which we won't have time to discuss it in detail. Oh, thank you. So the short answer, and before answering, I have to kind of decode what you said for the rest of the audience, because it turns out there is another pathway in the cell, which is not the N-degron pathway, but the pathway that involves ubiquitination, N-alpha terminal ubiquitination of proteins. It involves relatively small, but probably dozens at least, of different proteins. And of course, there is only one alpha amino group. So if you N-terminally ubiquitinate something, you cannot recognize it by the N-degron pathway. So there is potentially a competition between recognition by the N-degron pathway and recognition by the pathway, which is still, by the way, not very well defined mechanistically, that places ubiquitin moiety at the alpha amino group. They are now emerging. I'm not aware of clear understanding of this system, but they are now emerging specific sequences, kind of internal degrons that are positioned close to the N-terminus of the protein, that dictate, confer on a protein the ability to be N-terminally ubiquitinated. And relationship between the N-rule pathway and the system that you mentioned is going to be very interesting, but it's not very well understood, not by us, not by anybody else.
Take the last question because you need time for lunch before going to the Cooper. In many of your slides, you have amine as a regulator of different enzymes.
Yes.
It's amazing because for years, amine was just considered as being the prosthetic group of metal enzymes.
That's right.
And amine is supposed to be redox active when it's not conjugated to any protein. So what is your role, your feeling about the role of amine?
We actually discovered this about 8 years ago. And I was trying to convince, ever since I was trying to convince new people in the lab to take it on and deepen it. And as you probably know, happens very often, given the flux of projects in the lab and the limited size of my laboratory, we were actually not able to deepen this understanding beyond the fact that heme definitely binds both here, which is also a surprising thing to me. Why to bind at 2 different steps in the pathway? Because it binds here and changes properties of ubiquitin ligase. It binds here, has micromolar affinity for arginine transferase and inhibits it completely. And moreover, it increases the rate of degradation of AT1. That is, arginine transferase is also destroyed by him. So that it downregulates it at 2 levels, by preventing its enzymatic activity and by increasing the strength of its already naturally present degron. And so the partial answer to your question is that heme, or in this case hemin, ferrum 3 version of heme, is actually a very toxic molecule. It creates all kinds of oxygen radicals. It does other things. Therefore, cells evolved chaperone proteins to handle heme. In fact, the concentration of free heme in the cell is of the order of picomolar. It's very, very low. Right. And then these carrier proteins, so chaperone proteins for heme, donate heme to where it belongs. At the same time, heme is known to be circadian regulator. It does other things. Because we know already that the N-rule, I didn't mention it, I didn't have time, the N-rule pathway, for example, is a circadian regulator, among other things. It regulates circadian rhythm. And heme regulates circadian rhythm. So we anticipate once somebody, either in our lab or somewhere else, takes up this finding and makes it actually mechanistically explicit, what might emerge, to give you an example, is a relationship between heme dynamic in circadian rhythms and the function of the N-rule pathway in regulating those rhythms. Beyond that, we don't know.
Thank you. Merci beaucoup. So it has been a real pleasure to have you in this room. Thank you for your lecture. Thank you for the discussion and your answer. And we will see you under the coupole.
Yes. Thank you.
Merci.